Though there is little debate as to whether stem cells are a useful tool in medical research and treatment, it has been difficult to avoid the ethical controversies that surround this issue. When a stem cell divides, it can either reproduce another stem cell, or it can become specialized. This means by collecting and manipulating them, we can get a vast array of functioning specialized cells. This ability allows stem cells to have regenerative properties as they can replace many types of worn down or damaged tissues. One type of stem cell is the adult or somatic stem cell. When dividing, these adult stem cells are able to self-renew into any type of cell in the organ it originated from. In addition to the somatic stem cells, there are also embryonic stem cells. Embryonic stem cells are pluripotent, meaning they are able to differentiate into a cell of any type of organ it pleases. There has been a lot of controversy on gathering these cells, however, because the main way of getting them is through human embryos. Embryonic stem cells are taken from the inner mass of a blastocyst, which is formed between 4 and 5 days after fertilization. Removing stem cells from a blastocyst could damage or kill the embryo it was taken from. However, stem cell research is typically done using eggs that are fertilized in vitro. This is a process in which the egg is taken out of the woman's ovary and the embryo is grown in a petri dish in the laboratory. The blastocyst is typically only considered to be a mass of cells. The ethical debate here lies between when an embryo actually becomes a living human fetus and whether it is morally acceptable to remove these embryonic stem cells.
Due to moral objections to the harvesting of stem cells from human embryos, researchers attempted to find an alternative viable method to procuring stem cells. A key progression was found in work on both embryonic and adult skin cells in mice. In 2006, Kazutoshi Takahashi released his famous paper showing that it is possible to reprogram cells such as adult fibroblast cultures in mice to a state in which they are not yet fully differentiated. Mice cells were injected with nuclear contents to cause the induction of pluripotency. The amazing aspect of these newly derived stem cells was that they seemed to mimic embryonic stem cells in morphology and function. These cells were named induced pluripotent stem cells, or iPS cells, by their research team. While iPS cells were first researched in mice, it was later discovered that it was possible with humans as well. It remains uncertain as to how effective iPS cells can be in repairing damaged tissue in humans. A major impasse to return differentiated cells to an undifferentiated state is the use of a viral delivery system, which sometimes causes harmful effects such as cancer in animal test subjects.
In early 2014, a Japanese research team at the Riken Center for Developmental Biology claimed to have discovered an even simpler method of inducing pluripotency in stem cells. This method, called stimulus-triggered acquisition of pluripotency, abbreviated to STAP or STAP, involves subjecting fully differentiated cells to external stress in order to return them to a pluripotent state. A paper detailing the process was published in the scientific journal Nature in January of 2014. Haruko Obokata, a biologist at Riken, tested multiple stressors and found subjection to low pH in combination with a bacterial toxin that perforates the cell membrane and physical squeezing to be the most efficient way of performing STAP. In order to demonstrate the pluripotency of STAP cells, the next step is to tag them with a fluorescent marker and implant them in an early mouse embryo. If the cells are truly pluripotent, the fluorescence will be seen in every tissue of the mature mouse. After not having much luck with this test, Obokata started stressing differentiated cells from newborn mice instead of adults. This worked to demonstrate pluripotency. STAP cells are capable of forming placental tissue. This is impossible with other iPS cells. This means that STAP cells have the potential to be incredibly useful in cloning. If STAP cells can create their own placenta, it can be inserted directly into a surrogate. This could be more efficient than the current method of extracting unfertilized eggs, transferring a donor nucleus to the egg, cultivating an embryo, and then transferring the embryo to a surrogate. Stimulus-triggered acquisition of pluripotency is much more efficient than the previously discovered methods of inducing pluripotency. Dr. Takahashi's method has a successful conversion rate of only 1%. Haruko Obokata has stated that her method's STAP conversion rates are around 7.5%. If true, this is a 750% increase in efficiency. The new method is also much quicker, taking hours rather than weeks. One final benefit of this method is that it doesn't have detrimental effects on animal test subjects like using viruses to induce pluripotency does. Since being published in January of 2014, Haruko Obokata's paper has come under much scrutiny. After many failed attempts to reproduce her findings, the Riken Institute launched an investigation into her work. As of April of 2014, Riken's investigation found Obokata guilty of research misconduct, though they also stated that this does not necessarily refute her findings. Whether or not Haruko Obokata's method will become a common way of inducing pluripotency remains to be seen. This method will likely be refined and modified until scientists are able to reproduce it with much more consistency.